Welcome back, students. Now in this section I'll be talking to you how mRNA is going to be regulated. How are the different ways in which mRNA can be regulated. So there are different what we call as post-transcriptional processes of the mRNA which can help to regulate gene expression by altering something with the mRNA itself. So let us look one thing, that is the splice site choice. Suppose there is one mRNA and it has many exons and introns. Okay, now the introns have to be removed and the exons have to be spliced. So I'll just write to you the exons that are there 1, 2, 3, 4. So if we have an alternative splicing site, what can happen is, if there is a different splicing site, what can happen is this 1, 2 and 3 might combine together to form one protein. So 1, 2, 3. Or the splicing can be changed such a way that it might be another protein that is formed in which it will be 1, 2 and 4. So what has happened? This protein is different from this protein. And what has been changed here? There is an alternative splicing that has taken place. Just by changing the number of intervening or the exons which are going to be spliced together, those can be changed. So this is known as the splice site process. So over 60% of the 30,000 genes in the human chromosome undergo nearly 60% undergo differential splicing. So that is how we can explain that these 30,000 genes give rise to 100,000 proteins. Now, other than alternative splicing, alternative polyadenylation or alternative transcription start site, all these are also examples of different way in which mRNA can be regulated at the level of the splice site. Splicing can be changed. And this is the reason, for example, tropomyosin, there are 7 tissue-specific isoforms, all because of alternative splicing of its pre-mRNA. So this mRNA has undergone alternative splicing and resulted in a different protein, maybe an isoform also. Now there is one more way in which mRNA can be regulated, and that is known as mRNA editing. What do I mean by that? The mRNA has been fully processed. It is all the introns have been removed and the exons have been spliced. The intervening sequences have been removed and the exons have been spliced. Now what happens? It may undergo additional post-transcriptional modification. Now one classic example for this is a fully processed mRNA with its 7 with a 5' cap. It is having the polyadenylate tail, poly-A tail. Everything is over. This mRNA has undergone splicing. Everything is over. Now what happens is this mRNA is ApoB100 mRNA, ApoB RNA. Now this ApoB RNA, one small change happens. So we have codon known as CAA at the 48% of the whole protein. This codon CAA gets converted into U. Minute C is converted to U, what happens? This is mRNA editing that has taken place. This mRNA editing takes place in the intestinal cells but not in the liver cells. Minute it forms C is converted to U. This indicates that it is a stop codon. So what will happen? The protein that is formed with this will be different, with this it will be different. So with this it will result in ApoB100, which is the full protein, whereas with this it will stop at this level because there is a formation of a stop codon and it results in the formation of ApoB48, which is 48% of the whole ApoB100. So that is the reason which we call it as mRNA editing. Editing has taken place in the intestinal cells. So these are known as additional thing. A base in the mRNA may be altered and this is known as RNA editing. So as I said, formation of B100 and B48. Now there is one more thing by which mRNA can be regulated, and that is known as mRNA stability. So how much protein is formed depends on how long that mRNA is in the cytosol before this mRNA is going to get degraded. How long is this mRNA going to remain in the cytosol? That only will tell whether it is going to form, how much of protein it is going to form. Now, one of the classic examples for this is given as the transferrin receptors. Now, there is this transferrin receptor. At it has certain stem loop structures, what they call as stem loop structures in its mRNA. Okay. Now, the stem loop structures are present in its mRNA. If iron is there, or there is what is called as iron response protein, I'll just show this protein as like this. Now, if this protein it has a binding site. These stem loop structures have a binding site for this IRP. I call this as IRP, iron response protein. Now, when iron response protein is added to it, when iron response protein comes and binds to it, what will happen? It will increase the stability. It will increase the stability when with these blue things, this mRNA becomes more stable. Okay. So this stem loop structure IRP will come and bind to the stem loop structure, and it will increase the stability of this mRNA. This is transferrin receptor mRNA. Transferrin receptor mRNA. So this mRNA could have been degraded. So what will iron response protein come? It won't be degraded, and what will happen? Transferrin is produced. Transferrin receptor protein is going to be synthesized if this mRNA is stable. Now, when the body iron stores are good, this iron will go and bind to this IRP. Iron will go and bind to the IRP. When iron goes and binds to this IRP cannot come and bind here. When IRP cannot come and bind here, this is iron. What will happen? When IRP does not bind, what will happen? This mRNA gets degraded faster, and thereby there is no transferrin receptor formation. So when iron stores are good, we don't want transferrin receptor. We don't want to the cell doesn't want to take in more and more of iron. So it wants to degrade the transferrin receptor mRNA. So this is known as the transferrin receptor and the iron status, how it regulates within the cell. So this is a regulation by affecting the mRNA stability. Now there is one more procedure known as by mRNA can be regulated, that is RNA interference. This is also called as RNA silencing or RNA inactivation. This is by the formation of a double-stranded RNA, and this double-stranded RNA is recognized by dicer or endonuclease, and it is cleaved into smaller molecules called as short interfering RNA, siRNA. siRNA associates with proteins to form an RNA-induced silencing complex or RISC, and RISC hybridizes with the target RNA, and a slicer in RISC degrades the target RNA. So again, this is a big procedure. This is known as RNA interference, and this might be a part of your MCQ, and hence I am talking about all this. RNA interference is supposed to be a part of the body's defense system against retroviruses. So minute we use the word HIV, we need to know how HIV, everything related to HIV. So RNA interference is supposed to be one of the body's defense system against retroviruses, and this is how it acts. Now, regulation of gene expression can also occur through modifications to the DNA. Maybe how to access DNA, the amount of DNA, arrangement of DNA, or what are called as mobile DNA elements. I'll just tell you briefly about this. First is the access to DNA. I'm not going to draw anything. I'm just going to please look at the slides and try to understand what is happening. The chromatin fibers of the chromosome can exist in a highly coiled state, and this is called as heterochromatin, or in a relaxed state called as euchromatin. It can be highly coiled or it can be relaxed. So this is known as hetero or euchromatin. Now genes are found within the heterochromatin are not expressed. So one of the classic example is the bar body. It's a chromosome that is entirely in the heterochromatin form. The bar body is a chromosome that is entirely in the heterochromatin form. In other cases, smaller regions of the chromosome may become heterochromatin in some tissues and euchromatin in other, and thereby different tissues are able to regulate the levels of different proteins. So here the access to the DNA is altered either by heterochromatin or euchromatin. Why the name? It actually depends upon the staining characteristics, but more than the staining characteristics, it is the genes which are involved and all that, whether it is highly coiled or in a relaxed state. So that is why I have not gone for the staining, what changes takes place. Okay. Now further, there is something called as chromatin remodeling. So conversion of active and inactive forms of chromatin takes place. This is mainly by what is called as acetylation of the lysine residues at the amino terminus of the histone proteins. So we have histone proteins associated with DNA. Let me just tell you about it. You have a huge histone DNA, and this DNA is coiled and coiled and coiled, and it is associated along with histones. Now what will happen is the histone protein has a lysine residue. Please look here. This lysine residue can get acetylated. What brings about this acetylation? And when it is acetylated, what happens? There is decreases the interaction with the DNA. The histone proteins which are supposed to interact with DNA. Why? Lysine is a positive amino acid which is positively charged. It reacts with the negative charges of the DNA and sees that the DNA is closely associated with the histone protein. But minute this lysine residue gets acetylated, the DNA does not react so closely with the histone protein. So this opens up the chromatin and this helps in transcription. Now there is an enzyme known as histone acetyl transferase, which is known as HAT. HAT is histone acetyl transferase, which causes acetylation, and this is reverse is done by histone deacetylase. This is one more mechanism in which DNA is going to be regulated. Another mechanism is again access to DNA is by extent of methylation of the cytosine in the GC rich regions in the upstream of many genes. Again we are looking at methylation. Methylation is done with the help of S-adenosyl methionine. So transcriptionally active genes are less methylated. Methylation ensures that the genes are turned off and remain turned off. Okay, that is the importance of S-adenosyl methionine. So regulation of gene expression through modification to DNA, as I said, access to DNA, amount of DNA can be changed, arrangement of DNA, transposons, mobile gene elements, so many things are there, mobile DNA elements are. So with this we come to the end of our how genes are regulated. I'll just summarize to you how genes are regulated. Within the body by eukaryotes. So you have got at the level of you have the DNA. So genes can be regulated at the level of transcription and formation of a mRNA. Secondly, you can control at the level from this mRNA is going to leave the nucleus and it is coming out. So this mRNA processing and forming the hnRNA to mRNA at this level. So first level could be DNA to mRNA, mRNA to hnRNA to mRNA, DNA to hnRNA, hnRNA to mRNA. Further, mRNA is converted into different levels, different things, modifications can happen. It can be converted into a protein mRNA stability itself. So the third level will be at the level of mRNA stability and mRNA processing, you can say, mRNA editing, etc. all at that level also. This mRNA is going to form going to form a protein. So at this level also, these protein and proteins things that can happen, those also can be regulated. How much of protein, mRNA stability, etc. protein. Then further regulation can occur at how this protein is going to change. So this and at how after how much time this mRNA is going to get degraded. So that also can be regulated, and this post-transcriptional modification, post-translational modification also. So modification of the different enzymes, different proteins. At that level also we can regulate. So these are the different mechanisms by which regulation of gene expression occurs in the human being, occurs in the eukaryotes. There are so many things involved in this. So with this way, I hope you have understood, and especially one important thing that needs to be taken home is RNA interference, which is the body's defense mechanism against HIV or retrovirus. So in that there is a formation of disc, risk, etc. But just remember RNA interference. Many drugs are being tried, so especially for hypertension and all that. Can we interfere with the RNA of the different proteins which are responsible for forming a different protein? RNA interference, in which another compound or another nucleotide is trying to give and see that it interferes with the conversion of mRNA into a protein. So with this we come to the end of regulation of gene expression by different mechanisms. Thank you.